Well, thanks for coming this early, the last day of the conference. I am indeed not Mark Bullrich, as it was announced. I am Diego, and I'm going to tell you about large-scale brain networks dynamics, different modalities and time scales. So much of what's been discussed today is about average network activity. Average network activity is a great thing to look at because it's a really strong, robust phenotype, subject-specific robust phenotype. It tells us a lot about the functional architecture of the human brain, but it's not unreasonable to assume that there is more information above and beyond this average. Thank you. So we know actually that this average is composed of different networks of brain activity that have distinct dynamics. And I'm going to talk today about how these different networks appear to us across different time scales and across different modalities, how they are similar or dissimilar across these time scales, and I'm going to end up trying to conceptualize or make some sense of these similarities or dissimilarities. But first I'm going to start with a little bit of methods discussion about how we measure and how we assess and quantify these networks. So the way people normally look at dynamics in this network activity is by the use of a sliding window. So you basically choose some width of the window, you look at the network activity within that window, then you move the window and you assess this network activity again, and then you have a moving picture of this connectivity. There's a number of issues with this approach. First is there's not an obvious way how to choose the width of the window. You choose it too short, then you get an unstable, unreliable estimation of the network dynamics with changes that might be spurious or actually a consequence of just noise. If you choose it too long, you miss on quick things, obviously. So at the end, time resolution is limited by the size of the window itself, and also by doing things that way, you make an inefficient use of the data, which manifests in these problems with having a noisy estimation. So what we normally do, or we like doing, is to change this approach and look at a different perspective, which is based on brain states. So we describe the time series. We assume that the time series can be reasonably described using a discrete number of states, so that each state can on and off sometimes, and they recur over time, so that we can pull from these occurrences to estimate each of these states with a lot more data than you would be doing with a sliding window. That obviously doesn't mean that we believe the brain has that discrete number of states. So this is just a description of the data that can be useful for some purposes, or maybe not for others. But it's at the end of the day a description of the data and a data analysis tool as ICA or PCA will do. So the framework that we use is called the hidden Markov model. It's a probabilistic framework that gives you an estimation in one go, in a data-driven way, of when these networks or these states occur, which are the parameters of these states, which tells you information about, for example, connectivity in this case, and also a transition probability matrix, which tells you which is the probability of going from one state to another. So it sort of governs the dynamics of these networks. So one of the nice things about this approach is that it's a generic framework that you can adapt to different modalities by using different, so to say, descriptions of what a state is. So I'm going to start with a few examples of results that we have with fMRI, and then I will show some other results with other modalities, and we'll see how these are different or similar. For example, with the hidden Markov model and using resting state data, we were able to describe in quite detail the spatial properties of the different resting state networks, and we also found striking properties on the temporal activation. In particular, we found that these networks temporally organized into different groups we called metastates, such that the probability of going from one network to another within one of these groups is much higher than across groups. We did something very similar in sleep. We again found this hierarchical structure of these temporal dynamics of these networks, although here these metastates were actually quite well mapped, or matched quite well what we know about the different stages of sleep. So that's with fMRI, where we have a lot of, as you know, a lot of spatial resolution, and we want to look at MEG, which is from a temporal perspective, is much richer, has information across different frequency bands. We know underpin different functions and have different properties. In particular, we were interested in phase coupling, phase locking. So two signals phase couple when they sort of are in phase, and this is thought to underpin actual communication between neural populations. So two neural populations are thought to actually communicate when they are actually in phase. That's something people have been investigating in the visual system and being quite specific in space. What we wonder here is whether or not there are maybe transient networks of phase coupling across the whole brain. For this, we used a different version of this hidden Markov model business, where the states were defined by some patterns of autocorrelation across regions, but also across time, sort of a truncated Gaussian process, if you like these sort of things. So the critical thing here is that this description of the states give us information of power across different frequency bands. So we have a description of a spectral amplitude, so to say, for each frequency, and we also have information about coherence or phase coupling. So we pulled out a number of networks or states from, again, resting state. I'm showing here two of these that we found particularly interesting. So each of those is defined as a pattern, as I say, of power or amplitude, and a pattern of coherence, and that is defined across frequency bands. So I'm showing here two frequency bands. These frequency bands are, we call them delta and alpha, although they don't look exactly as you normally delta and alpha look. That's because we did sort of a data-driven factorization of this frequency information. So this came out of the data in a sort of, automatic way. So we see these two networks. One of them we call anterior DMN, or anterior higher order cognitive, or I think that's the term we use in the paper, and the other one is a posterior DMN. So we see that the anterior DMN has a lot of power in frontal regions of what is normally considered DMN, and also temporal poles, and has critically a strong phase coupling between the PCC, which is a region that is normally not that easy to see in MEG, and these frontal regions in these slow frequencies, these delta frequencies. And also in alpha has more angular gyrus and also a little bit of PCC. The other one instead has no remarkable features in the slow frequencies, but it has also power and phase coupling with the PCC and also the angular gyrus. So it seems like the DMN is sort of split into two different components. And indeed, this is exactly what happens. So you only see the actual DMN as we know it from fMRI if you get the union of these two networks. So it seems like MEG, which is looking at much different time scales, much quicker time scales, is putting out something related, but still a little bit different from what we see from fMRI. So MEG networks have dynamics of fast time scales, and we wonder whether these MEG networks have dynamics at slower time scales that maybe match a little bit closer what we see in fMRI. So in order to address this question, the obvious thing to use is simultaneously recorded EEG and fMRI data. So the pipeline we used here was we got some EEG data, we ran the HMM on that, so we have state time courses of each state, so probability of being active at each time point for these states. Each of those is a network in EEG. So we convolve these time courses of probabilities with the HRF, and we downsample, so now this has the same scale and resolution than fMRI. And then we use a standard GLM on the fMRI data to pull out networks from the fMRI data, but the time scales that we got from this, not the time scales, but using the time courses that we obtained from EEG, but at the time scales of fMRI. So we have EEG HMM networks from fMRI and EEG, and we compared those with the classic ICA, group ICA fMRI networks. That is basically what most people do, and we found, or Body found, that that these were actually quite similar. So there's an intrinsic relationship between EEG and fMRI in this case. So the hypothesis, or the way we conceptualize this, is that perhaps the resting state networks that we see in MEG sort of burst, and they go on and off, but these bursts, when looking from a little bit farther away, from a slower time scale, maybe burst at time scales that match with fMRI. And perhaps if we were to look at these MEG networks, the BMN2 networks that separated, maybe if we were looking at a slower time scale, they would be seen as we see them in fMRI. So this is in line, for example, with work from Dimitri and Deville, for example, with EEG microstates, where they also looked at different time scales and how these time scales relate to each other. And with a little bit of time, I'm going to finalize my talk. And well, you see this. I think this, from a methods point of view, I think it's useful to have a bag of different lenses, and in particular, having a framework that can be used across time scales is useful for interpretation purposes. And in this case, we've seen that there's fMRI networks that have rich spatial properties, but also temporal properties that are remarkable. And we have quick MEG states that fluctuate quickly. They have specific properties in terms of phase coupling, which we were interested in. And the critical point of this talk is that they sort of burst these MEG networks, or these EEG networks, or electrophysiological networks. They burst time scales that actually match quite well with what we see with fMRI. So at the end of the day, it seems that we see things with fMRI and EEG that are common. Obviously, there are differences, but not everything is random, guys. And that's it. Thank you very much. I just noticed that this thanks slide is a bit old, so I'm missing people here. But so I apologize for those who are not in the thanks slide.
Thank you.